Good morning, class. And thank you, Caitlin. And thank you, Rupa, for inviting me to speak on behalf of Cyrex about the subject which is so close to my heart. And this subject is very well summarized in two articles that I published in 2021 in journal Pathophysiology. I'll read the title for you. That the first article is about the role of exposome, whatever we are exposed on daily basis, in pathophysiology of autoimmune diseases. The first one mainly is about food and toxic chemicals. The second article is about the role of pathogens in autoimmunity. So if you would like to read, these are the two major articles supporting my presentation. So in those articles, I did emphasize the importance of genes plus environmental factors, food, toxic chemicals, pathogens, and their effect on the gut microbiome that may induce autoimmune disease and other immune disorders. So let's start with food. In relation to food, we at Cyrex, we believe in food-associated autoimmunities. So when a laboratory is measuring IgG or IgA antibodies, should not claim this is about allergy. I call that food immune reactivity that may contribute to autoimmunity. The real allergy is if you measure, or if the laboratory measures antibodies, e-antibodies against food. Food-associated autoimmunity is because many foods share homology or cross-react with human tissue through different mechanisms, including molecular mimic and autoreactivity, which contribute to inflammation and autoimmune disease. So I would like to share with you the major factors which are important in reporting reliable test results, because the accuracy of any lab tests depends on purity of the antigens used in the test development. So if the food, proteins, antigens used in the assay development are not pure, you are going to get either false positive or false negative results. That's why at Cyrex, we spend years of purification of our antigens and applying that pure food antigen in our testing, which is giving us highly reproducible and reliable test results. The next to that is optimization of antigen concentration. Let me give you one good example. Let's take apple or parsley versus soy or peanuts. Soy and peanuts have protein concentration of around 20 to 30%, while apple and parsley, their protein concentration is less than 1%, probably 0.1%. So how come laboratories, they take similar volume of extract from apple or parsley versus soy or peanuts, apply that to their testing, and they measure antibody again? That's not right. We do not call that optimization. The lab should use the same concentration of protein, whether it is from parsley, apple, or soy, or peanuts. If they don't do that, in one case, in the case of peanuts or soy, may get false positive results. In the case of apple and parsley, may get false negative results. So we call that antigen optimization. Also, these days, through many publications, there are lots of synthetic peptides and recombinant proteins are available, which we prepare those through different companies, which is their specialty, and use that in our assays. Let me give you two examples. Kinase is an enzyme found in rice. So you can test for rice, but if you don't test for kinase of rice, which is highly purified and synthesized, you may get false negative results. So the only way you get accurate results is not only to test antibody against rice, but also to test antibody against rice chitinase. That's what we do at Cyrex. Second example, pineapple. You can take extract of pineapple, measure antibody against that. But that doesn't mean that you are reacting or you are not reacting to bromelain. So you have to take bromelain separately, highly pure, apply that to the testing and measure antibodies against light. That's what we do at Cyrex. Next, and the most important, is raw versus cooked foods. And I'll show you lots of evidence and share with you even an article published a couple of months ago in journal Foods that individual may react to raw food, but not to cooked food, and vice versa. One individual may react to cooked food, but not to the raw food. And classical example is egg. We know the temperature changes the protein structure of the food. And so therefore, change in the protein structure does not reflect prepared from raw food. So why almost all the laboratories other than Cyrex testing antibodies only against raw food, but not against cooked food, which we do both at Cyrex. Lectins and agglutinins are highly inflammatory. We measure antibodies against lectins and agglutinins, and highly important to know that lectins and agglutinins, for example, wheat germ agglutinin and soybean agglutinin, other agglutinins, cross-react with thyroid peroxidase. Therefore, if you have a patient with thyroid autoimmunity, you may look at possible immune reactivity against lectins and agglutinins that we do at Cyrex Laboratories. We measure antibodies against found in oils and nuts, and no other laboratory is doing that. Gum are highly antigenic. Their molecular structure can run from 200,000 to 5 million. Originally started using them in human diet, they thought they will be in and out, but that's not the case. Our digestive enzymes can digest the gums to smaller molecules, and then immune reaction against them results in antibody production, and those antibodies can cross-react with human tissue. In fact, we have seen that gums and immune reaction to gums may result in polyreactive antibodies, meaning an individual not only reacting to gums, but that antibody may react with additional 10 or 20 or 30 different food antigens and different human tissue antigens. Artificial food color actions. Antibody measurement against those is possible only if we combine the food coloring to a protein like human serum albumin, and then we measure antibody against that. Unfortunately, some laboratories do not have this knowledge. They don't understand the new antigen formation. They just take artificial sweeteners, add them to the test tube, and they measure antibodies against artificial sweeteners, which is impossible to do. So therefore, to measure antibodies against chemicals, including artificial food colors, you have to bind these small chemicals to a protein and then measure antibody against that. That's what we do at Cyrex. And then finally, there are cases that many food components bound together and we measure antibody against that. One of the best examples of that is different foods combined together through meat glue or microbial glutaminase. An example of those you can see in here. Ladies and gentlemen, the picture you see in here, they are not real meats, they're modified meats, which taken smaller pieces of meat, taking the meat glue and stick them together and they look like beautiful steak. So we have to measure antibodies against meat and meat glue added to the meat in order to find out whether the individual reacting to the combination of these food antigens. That is done at Cyrex Laboratories. And then multiple antibodies, IgG and antibodies, as I said before, we do not make any claims about detection of IgG and antibodies against food in relation to allergy to food. The only claim we make is IgG and IgA immune reactivity may cross-react with human tissue. These antibodies may cross-react with human tissue and result in inflammation and immunity and not allergy. So one more time, I do emphasize that heat can change the structure of the protein as well as peptides. So different, then after heat, probably different peptides. The peptide changes, the protein changes, and so therefore it's completely antigen. So in summary of this part about the importance of reliable laboratory testing, it is important to measure antibodies against both raw and cooked food, but all commercially available food antigens are prepared these days from raw foods, whether in laboratory testing for antibody measurements or even for skin testing. So this exposure of food ingredients to these harsh conditions may cause alteration in immunodominant epitopes, potentially affecting their antigenic and allergenic properties. So Cyrex is the only laboratory that tests for both IgG and antibodies against raw and cooked foods prepared by myself through years of purification and other activities related to their preparations. So to support all that, I highly recommend to read this article published in journal Nutrition and Metabolism as early as in 2009. So we are the original inventor of food immune reactivity and autoimmunity. I did invent food IgG testing in 1986 or 1987. And through other activities, as you can see in here, throughout the years, I added to that development. I brought it to almost to close to the perfect testing. So in 2009, we measured IgG, IgE, IgA, IgM antibody against raw and processed food antigens. And we found that some individuals reacted to raw, some reacted to modified food, and some reacted to combination of raw and cooked food. And you'll see some examples of those. This is taken from the same article. Before publishing this article, I read an article which was published in one of the immunology journals. And that article, they extracted wheat from sausage and measured antibody against wheat extracted from sausage versus antibodies measured against raw wheat. Many individuals did not react against wheat flour, but reacted strongly to wheat extracted from sausage. I call that modified wheat proteins. So here, another example that some reacted to wheat right here, barley a little bit, advanced glycation end products a little bit, soy a little bit, but look at extracting antigens from donuts. So all these proteins, when became modified, many individuals produce IgA, IgE, or IgG antibody against them. So one more time, many times may react to modified food, but not to the raw foods, and some may react to raw food and not to modified foods. So this is the summary of that article. So as early as 2009, we showed that heat modification of food proteins and peptides results in the formation of new immunodominant epitopes or new antigens. This antigen formation results increase or decrease in the level of antibody production when it is compared to raw food antigens and antibody measurement against them. So many research studies have shown that individuals may not react against the primary or native form of a protein or peptide, but may react to their secondary, tertiary, and quaternary structure due to denaturation and even advanced glycation end products. That's what heat is doing to the foods. So this is the article published a couple months ago in journal Foods, available to you. You can go online to journal Foods and just put the title of this article, Immune Reactivity to Raw and Processed Foods and Their Possible Contributions to Autoimmunity. You can download that and read this article. In this article, we showed that classical example of egg, 3 minutes, 5 minutes, 7 minutes, and even hours, the protein structure changes from, you see, the primary structure, secondary, tertiary, and quaternary. Patient number 1, no significant reaction to raw and cooked egg. Second one, reaction to raw, but not cooked. And third patient, not to raw and cooked food. You see why it is important to measure antibodies against raw and versus cooked food simultaneously. One of the classical example for me is antibody measurements against chicken versus tandoori chicken. So raw in blue, red is cooked, and the third one is tandoori chicken, red, green, okay? So only about 4%, 5% general population reacted to raw chicken, IgG or IgA. About 6% reacted to cooked chicken. But when we purchased tandoori chicken and extracted antigen from tandoori chicken, which contains food colors plus proteins of chicken, now the percent reaction in the population went up to 13% from 5%. So you see the importance of food additives, their contribution to new antigen formation, and in here also measuring antibodies against wheat flour in blue, raw pizza dough in red, and baked pizza. You'll see some differences in immune reactivity against wheat flour versus pizza dough and versus baked pizza dough. Raw pizza dough in red and baked pizza dough in green. So the immune reactivity increased significantly for both IgG and IgA when wheat flour became modified. And this is summary of 808 individuals tested at Cyrex for raw versus cooked food, and raw is in blue, cooked food in red, and you can see significant differences in raw versus cooked food when we measure IgG plus IgA antibodies. So overall, there is more reaction against modified food than raw food, but there are some cases also, certain foods like banana, raw, higher reaction, and also grapes, higher reaction than banana cooked or red wine or white wine. So individual may react more to modified food, but some individuals also may react to raw food as well. I emphasized the importance of new antigens formation by chemicals, which are small molecules. Individuals may not react to small molecules, such as food colorings, and name of the chemicals that you can see in here, which are part of Array 11 or chemical immune reactivity screens, because chemicals bind to human tissue form neoantigen, and now our body react to chemicals plus human tissue antigens, and then that antibody combination contribute to immunity. So these are some of the antibodies we are measuring at Cyrex Laboratories. Aflatoxin bound to human serum albumin. Formaldehyde, glutaraldehyde, isocyanate, phthalic, benzene ring, bisphenol A binding protein, tetrabromobisphenol A, tetrachloroethyl, parabens, mercury, and mixed heavy metals. All of them bound to human serum, and then we measure antibody against them. At Cyrex, we do not take the chemicals like some other laboratories are doing. Directly add them to the test tube, and they measure false positive antibodies against them. We are measuring specific antibodies against chemicals bound to human serum albumin. We call those neoantigen formation, which is reflection of body burden of toxic chemicals. As you can see, I present these topics in International Congress of Autoimmunity, as well as publish that in a journal called Journal of Applied Toxicology. So we bound chemicals to human serum albumin, and we measured antibodies in healthy subjects. We found that 15 to 20% individuals that we tested did not react to chemicals by themselves, like measurements of antibodies against the artificial sweeteners, which is impossible to detect if they are not bound to human proteins. But when we bound those to human serum albumin, up to 20% of the population reacted to some of these chemicals, including bisphenol A, bound to human serum albumin. So that's the right way to do it. And to support, I would like to share some articles with you, not my articles. Just please look at the title, Protein Adducts Biomarkers, State of the Art. So covalent protein adducts formed after exposure to biotics may provide readily measurable indicators of these exposures. That's exactly what we did, because chemicals form antibody, form adducts with our proteins, with our DNA. By making antibodies against these adducts, we can detect them in the blood. That's exactly what we do at Cyrex. So chemicals cross-link to DNA or proteins cause significant damage to the DNA. Chemicals cause significant release of autoantigens, meaning our tissue antigens are released. And when our tissue antigens release, more chemicals bind to those released tissue antigens. Now, immune reaction against them results in inflammation and immunity. Autoimmunity, the antigen hypothesis. That's exactly what I was emphasizing in earlier slides. Progress in understanding the molecular pathways that generate neoantigens will enable the design of new preventive and therapeutic approaches, which could be applied prior to the preclinical detection of autoantibodies or once the disease has been established without interfering with normal function of the immune system. So if individual chemicals, antibodies detected in the blood, let's say it's formaldehyde bound to human serum albumin, you have to find the source of the exposure of the patient to formaldehyde, remove that from the patient's environment, and prevent development of autoimmune disease in the future. That is message of this fantastic article. So which one is more important, to detect chemicals in the urine or even in the blood, which all of us has? If we measure all those chemicals mentioned in the slide before, believe me, 99% of us will have those chemicals in our urine. But measuring antibody against chemicals bound to human tissue, that is indication of body burden of toxic chemicals. So I do not recommend measuring of chemicals in the urine or in the blood, but measuring antibodies against chemicals bound to human tissue based antigen formation mechanism. So this individual, for example, reacted to isocyanate found in paints, to trimellitic anhydride also found in paints and plastics, and also benzene ring found in plastics and many other chemicals. So practitioners, by removing these chemicals from environment of the patient, can prevent autoimmune diseases in the future. The antibody, also the individual reacted to tetraethylene, which is found in dry cleaning, and similar to that chemical is in fire retardant as well. So we have to be aware of that. So that was my message about chemical immune reactivity. You have to measure antibodies against antigen formed in human body due to exposure to chemicals and chemical binding to human tissue. The last part of my talk is about the pathogens. At Cyrex, we measure antibodies against 29 pathogens, from oral pathogens to bacteria, viruses, parasites, molds, Aspergillus, Penicillium, Stachybotrys, citrullinated EBV, Borrelia, herpes type 6, and others. And these antibodies measured against these 29 pathogens do cross-react with human tissue and contribute significantly to immunity. So pathogens, release of associated antigens, cause release of associated antigens, cause inflammation, breakdown in the barriers, activation of the immune system, T cell, macrophages, B cell, release of pro-inflammatory cytokines, which can result in inflammation and autoimmune. So the Cyrex difference is that we are measuring antibodies against pathogens that can cross-react with human tissue. So while other laboratories are interested only in infection, Cyrex focus is on infection that may play a role in autoimmune diseases. Example, Helicobacter cross-react with human brain tissue antigens. That's why Helicobacter antibodies are detected in very high percentage of patients with multiple sclerosis. So by finding that, you may treat against Helicobacter and prevent multiple sclerosis development in the future. And these are some of the examples. All those red amino acids shows cross-reactivity with myelin basic protein and with myelin oligodendrocyte glycoproteins involved in induction of multiple sclerosis. Innate immune recognition of molds and homology to the inner ear protein called cochlin in patients with autoimmune inner ear disease. You saw that Cyrex, we are measuring antibodies against Aspergillus, Penicillium, and Stachybotrys. That and detection of antibody against those three molds is the best indication of exposure to the molds or presence of Aspergillus and Penicillium, not the Stachybotrys, as part of human gut biome. Why that is important, this article shows that significant cross-reactivity between Aspergillus, specific peptide, with protein in human ear, which is causing so many problems. So Aspergillus niger, all the red amino acids shows cross-reactivity with cochlin. The same thing, Penicillium, cross-reactivity with human cochlin. You make antibody against Aspergillus or Penicillium, those antibody may cause autoimmunity against the ear tissue antigen. So by treating or removing the molds, you may prevent autoimmunity in the ear. And Epstein-Barr virus cross-react with lupus-associated antigen. almost 70%, 80% cross-reactivity. So if you treat for Epstein-Barr virus, you may prevent lupus immunity. So these are the message of Cyrex. We know that Epstein-Barr virus, due to cross-reactivity, contributing to at least 30 different autoimmune diseases. This is one of the example of pathogen-associated immune reactivity screen. This individual reacted to H. pylori, also reacted to Stachybotrys chartarum. Both cases are very easy to treat. You can give antibiotics to treat for H. pylori. You can remove the molds from the environment of the patient and prevent serious autoimmune diseases in the future. And finally, the new development at Cyrex called biome burden. The biome burden developed because especially the microbiome components of the gut, in addition to microbiome components, were ignored completely. So that's why I would like to give you a couple of slides and about this subject. So also I would like to share with you this article published in Cell Host and Microbes, published in October 2024. We have to pay attention to the microbiome, the molds, especially the candida, the fungi in the gut. So in the healthy gut, you see there are only few hyphae. And so there are some anti-candida antibody are produced, controlling, preventing from these hyphae to penetrate the tissue or cause damage to the gut barriers, our immune cells, by producing IL-17, and our Th17 can take care of extracellular microorganisms such as Candida albicans. But overgrowth of pathogenic bacteria and increase in hyphae formation, like in here, can cause damage the gut barriers, entry of unwanted antigens, release of interferon gamma, TNF alpha, and activation of fungal specific T cells can cause significant damage to the gut, resulting in inflammatory autoimmune disorders such as Crohn's, ulcerative colitis, and even all the way to the brain immunity. So that's why recently, after 2 years of research and development, I came out with these tests. We measure antibodies against 6 species of Candida albicans. Candida albicans is important, but you have to measure also antibodies against other species of Candida. Measuring antibodies against Candida and other species is important, but without measuring antibodies against virulence factors, you miss significant components of micro-involvement in inflammatory and immune disorders. That's why at Cyrex now we are measuring antibodies not only against 6 species of Candida, Candida-like, Candida germ tube, and alpha-E, which all contribute to Candida to become virulent. Saccharomyces from food, including Saccharomyces boulardii, fungi, Aspergillus, Penicillium, Cladosporium, Alternarium, and others do cross-react with Candida. So altogether, fungi and yeast can contribute to autoimmunity. That's why we are measuring all of that. That was about the microbiome role in autoimmunity. How about the aerobe? E. coli, Salmonella, Clostridium difficile, Perfinia indica, Bacteroides. Important to measure antibodies against those as well. Those are mainly our microbiomes causing inflammation and autoimmunity. The neutrophil's job is to protect us against all the above. When they cannot do the job, they release all these enzymes, and we measure antibodies against all these enzymes. Both yeast and bacteria cross-react with human tissue and contribute to autoimmunity. That's why we are measuring antibodies against all those bacterial and fungal antigen that cross-react with human tissue. Finally, the 4 important T cells, Th1, Th2, Th17, and regulatory T cells are measured as part of Cyrex biome burden because they are playing a significant role to protect us against candida, yeast, aerobic and anaerobic bacteria, and prevent auto. So some of these bacteria and yeast and candida, unfortunately, form biofilm in the gut, and therefore antibacterial and antifungal may not work against them. Furthermore, these biofilm, due to candida and anaerobic bacteria, may not be detected in the stool by doing a stool culture. Even if they are present, the bacteria and yeast present in the stool, that doesn't mean they are pathogenic and causing inflammation and autoimmunity. You have to measure the antibodies against these yeast, bacteria, and their virulence factors, and their cross-reactive tissue antigens as well. So they are not detected, biofilm is not detected in the stool, and only by measuring antibody against. And this is a classical example of one patient reacted to Candida tropicalis, parapsilosis, Candida auris, which is found in the hospitals, but now we see antibodies detected in the population against that, antibody against Candida lysine. So yes, this Candida is contributing to, this Candida is pathogenic because making antibody against those mycotoxins and cross-reaction with food-derived molds. And finally, neutrophil became activated to take care of the body, but in the process gives up, and we make antibody against all those enzymes. And finally, the individual reacted against cross-reactive tissue proteins such as actin, and even that patient also, the T cell was elevated. T helper 17 was elevated because T helper 17 role is to take care of extracellular microorganism. That's why T helper 17 is elevated. And you see why it is important to measure cell-mediated immunity in addition to humoral immunity, which is the antibody detection. So, in conclusion, that autoimmunity has 4 different stages. The healthy stage, which is the green zone, which I want all of your patients to be in that zone. Stage 1 autoimmunity, we call that silent autoimmune. They may have some antibodies, but no symptoms, no loss of function. We call that initiation of autoimmunity. So if not in the healthy stage, the green zone, at least I want your patients to be in the silent stage of autoimmunity. So when you measure antibodies against food, against toxic chemicals, against pathogens, including those in the biome burden panel, then you can treat for those and prevent progression of autoimmunity from initiation to promotion and progression. So promotion is stage 2 autoimmunity or autoimmune reactivity, elevated antibodies with symptoms, loss of function, but no severe destruction of the tissue associated with the disease. And finally, and unfortunately, if we'll not detect the antibodies, if we'll not treat them correctly, if we'll not use the right laboratory, which is not reporting false positive, will report specific testing, then you can prevent progression from initiation to promotion and progression, which is the late stage of immune disease, significant symptoms and signs, lab imaging, special studies associated with significant loss of function is detected. So in conclusion, please consider to detect autoimmunity at the early stage as possible by looking at the role of exposomes, including food, toxic chemicals, and pathogens in immunity. Thank you so much.
Welcome to Rupa Health, the best place to order, manage, and track results from over 30 different lab companies in one single place for free. It's going to take you under 2 minutes to sign up, and you can order any functional medicine lab for your client in under 30 seconds. Let me show you how it's done. So here's our beautiful interface. I'm typing in the name of my client, selecting the lab that I want to order for them, and hitting send. From there, Rupa and their amazing team handle the rest. They email the client, collect payment, and even offer an interest-free 3-month payment plan. We've also built the world's largest library of information about chronic health conditions, the lab tests that can help you find the root cause, and the evidence-based interventions that you can use to help people heal from them. It's called the Rupa Health Magazine. There, we have in-depth articles about almost any health condition you can imagine. And we give you step-by-step protocols that other clinicians have used to help their clients heal and that are verified by evidence-based sources. You should totally check it out, and it'll transform your practice. And we can't wait to see you. So make sure you sign up today at rupahealth.com.